The topic is natural cause and impact on daily life, and I will present some results from the IPA Erasmus MC Pompe survey. And I'm very happy to do that here today on the AMDA IPA meeting, because it was truly a cooperation between the IPA and Erasmus MC. And I think many patients who are also present here today also contributed and participated in this survey. This is just a slide to show the clinical spectrum. You know already a lot about it. But I will focus today on the late onset form of Pompe disease, because these were the patients that participated in our patient survey. So what is the IPA Erasmus MC Pompe survey? It started in 2002 as an initiative of the International Pompe Association and Erasmus MC. And we did that because both the IPA and our center thought that with enzyme therapy underway, more insight was needed in the late onset patient population, and with respect to severity of disease, use of medical care, and how to follow up these patients. And we also asked ourselves the question, if you want to evaluate the natural course of the disease, and if you want to evaluate in future the effects of treatment, shouldn't we be asking patients if they are better? That's also the title of a commentary in the Journal of Clinical Epidemiology. If you want to do outcome research on treatment and on natural course, you have to ask the patient's point of view as well. So that was also a goal of the IPA survey. We wanted to learn more about the impact of the disease on patients' lives, and we wanted to include the patient's point of view in the measurement of outcomes of the disease. So it's an ongoing international study using self-report questionnaires, and inclusion criteria are just a diagnosis of Pompe disease and an age between 2 and 99 years, so just all late onset patients. What did we use for kind of questionnaires? Baseline questionnaire on medical history and current status, a shorter follow-up questionnaire. We used 3 previously validated questionnaires on fatigue, daily life activities, handicap, and on health-related quality of life. So this is the response we got by now. We have a total of 314 participants from several different countries all over the world, and all the patient organizations affiliated to the IPA in all different countries took part in the organization of the study. And we recently started a follow-up of this questionnaire, and it's still in progress, but we already have 119 patients again participating. So we're really glad that so many patients were willing to help us to complete the study and to gather as much information as possible. There are 3 topics that I want to talk about today. First is natural course and clinical variability and the results of this from the IPA survey, disease progression, and impact on daily life. I'll just start with the slide giving you an indication of what patients participated. This is from 253 patients in the survey, and this is the age distribution, and you see that it ranges from 2 to 82 years, with the majority of people between 30 and 60 years. And this slide is just to give you some indication of what kind of results we got from these questionnaires and from the IPA survey. And it shows the natural course of the disease on a group level. Here you see the age of the patients, and on the left you see all kinds of things that might happen during the course of Pompe disease. And this is the mean age at which these events took place, the 10th percentile and the 19th percentile. And I just pick out a few things to give you an illustration. The first complaints of the patients who participated in our survey, mean age was 29 years, but some patients already had complaints early in childhood and others after 40th year of age. Also for problems walking stairs, you see a wide variation in when that starts. Start of ventilation, the mean age was 49 years, with already patients who needed ventilation very early in life, and after the 7th year of age was also a patient in our survey. And this slide shows some other aspects of the natural course of the disease. You see here the age of the patients in the survey, here the age at which they start using a wheelchair, and the age at which they start using ventilation. And this patient started using a wheelchair at the age of 50 years, and at the age of 70 he also needed ventilation. And this patient first needed a ventilator, and then after 15 years later, also a wheelchair was necessary. And these are two examples of also patients with a different course of disease, starting at a very different age. And as an example of the differences in the sequence of events, I like to show you a short video, if it works, of a patient who is 24 hours per day ventilated. If you ask him to walk, he is still able to walk quite a distance. So that is to illustrate that the sequence of events can differ between patients. Some patients first have severe mobility problems, and then the respiratory part comes in, but by other patients it's more the other way around. With the information from the survey, we got 255 patients that participated at this time. I made this graph. And that means that you have a large group, and you can divide in subgroups and see if you can find a trend in the course of disease. And what we did was we made a division in age groups: patients younger than 15 years, 15 to 30 years, 30 to 45 years, 45 to 60, and 60 years and older. And the white blocks is the percentage of patients needing a wheelchair, and the gray blocks is the percentage needing respiratory support. And what you see is that the percentage does not change much between the age groups. There were mildly and severely affected patients in every age group. The only exception is the group younger than 15 years, where the percentage of patients needing respiratory support was lower. And we wanted to study this group into more detail, because when we looked at it closer, we found that the patients who did use respiratory support already needed it for 24 hours a day. They also needed a wheelchair, they needed tube feeding, they had early first complaints and an early diagnosis, which means there was a subgroup in the group of patients younger than 15 years with a more rapidly and severe course of the disease, and it was about 25% of all patients under 15 years. So what we could conclude, well, the first conclusion will not surprise you. Pompe disease is a genuine spectrum. First symptoms may appear at any age, and there are mild and severe patients in every age group. We also found that mobility and respiratory problems may exist separately, and that is important information to know also for your doctors, because it means that also patients who are still quite mobile need to have regular checkups of their respiratory function, because it may change independently of their mobility. We found a subgroup of patients under 15 years with a more rapid course, and that was about 25%, and these patients really need very regular follow-up to be able to intervene in time. So the results from the Pompe survey showed the large clinical variability in late onset Pompe disease. Then the question is, what determines this clinical variability? And you may say, well, perhaps it's the mutations that cause this variability. And by now there are over 200 mutations that have been identified in the GAA gene. And what I want to focus on is this mutation. It's for short called the IVS1 mutation. And it's present in 70 to 86% of all adult patients. It is a splice site mutation, which means that there's some residual activity left. And here I want to show you few patients with the same IVS1 mutation and a second mutation that leads to no residual activity. So these are 4 patients which have a comparable set of mutations, but still a very different clinical course. For example, here you see a boy of 4 years old here, and he already has symptoms of the disease, having difficulty standing up. sorry. And here you see a man of about 30 years who has no visible symptoms of the disease, and he has the same set of mutation as the boy I just showed you. Yeah. So what we want to know is, are there any things that can explain this variability in patients with this IVS1 mutation? It's the most frequent mutation in late onset patients. And they all have fully deleterious second mutations. But why I really want to show this today is because we were able to get some information from the IPA Erasmus MC survey on the natural course of the patients with the IVS1 mutation in our mutation database. So we asked the patients who participated in the survey, who had the IVS1 mutation, if we could use the information from the survey to link it to the mutation and enzyme activity. And luckily they agreed. So we had some information on the natural course in the study for about half of the patients with the IVS1 genotype. So when we were looking for factors that might explain the different courses of disease, we also wanted to look in the IVS1 haplotypes, and I'll try to explain what that is. First thing you have to remember is that all people are different, and they can also differ in the gene coding for acid alpha-glucosidase. And we looked at 17 places of the gene in which people can differ from one another. And also people who do not have Pompe disease differ on these sites. So it's not pathogenic, but it's kind of the background of the gene. And if you have so many different places that can differ between people, you can imagine you get a lot of possible combinations. And I will not go into much detail, but what is important here to remember is that 68% of the patients in our study with the IVS1 mutation had this haplotype, which means that the differences in haplotypes could not explain the differences in between the patients that we found. So the phenotypes and the haplotypes do not correlate. And this is also a slide from the study that I want to show you, just to show that what kind of information you get when you link the information from the survey to mutation database and activity. These are two patients with the same haplotype, that's a number, and the activity is different. This patient had an HR diagnosis at 1 year of age, this patient at about 30 years of age. But we found that the amount of activity is not really helpful in explaining the natural course, because these patients had also the same haplotype, also large differences in age at diagnosis and age at onset also. I didn't show you here. But the correlation with the activity was just the other way around. So the conclusion from this study was that secondary factors substantially modify the course of disease in patients with this IVS1 mutation. And yesterday we already heard a lot about secondary factors and phenotype. Nutrition might play a role, exercise might play a role, but also growth, maybe fiber type, other genes involved in glycogen metabolism. And I will not go into detail here, but I wanted to show you this to see what happens when you can pool all this kind of information, including the information that you provided in the IPA survey. So I will now go back to the primary goal of the IPA survey, disease progression in late onset Pompe disease and how to measure it. The slides Ans showed you yesterday, it shows increasing percentage of wheelchair use and respiratory support with increasing duration of disease. You see a gradual increase over time. And we also wanted to know if we do prospective follow-up of patients, whether we would be able to see differences with our questionnaires. And we did a pilot study in the Dutch participants, a 2-year follow-up. And here you see a graph plotting the number of hours ventilation per day against the time. This was the baseline measurement after 1 year and after 2 years. And you see that it increases, a statistically significant increase over time. And that was something that really surprised us because, well, late onset Pompe disease is known as slowly progressive. And And we were not really expected to be able to find in 2 years with these questionnaires already this difference. So it was quite surprising and in a way also quite shocking. This is the Rotterdam Handicap Scale. It was the scale we used to measure daily life activities, and you get a score. It is 9 questions on daily life activities, on mobility, on what people do in their leisure time, what they are able to do, work and study, household tasks. And of these 9 questions, you get a score between 9 and 36. And the higher score means a better function. And what I've plotted here was here the baseline score, and here the score after 2 years. And the patients who are on this line, here nothing changed. These patients scored better after 2 years, and these patients scored worse. And you see that the majority of patients is below the line. And also when you look on a group level and you do a statistic analysis, you see a significant decline in 2 years' time. Now this is not from the survey, it's from my colleague Nadine van der Beek, who works in the Department of Neurology. And she plotted the MRC sum score against the duration of disease. The MRC sum score is a measure, it's a manual muscle test performed by a neurologist. And you get a grade of the muscle strength of the patient. And you see here that it shows a gradual decline over time. And of course it's very important to measure muscle strength because it gives you a very good idea of the muscle strength of the patient. But I think what we showed with the Rotterdam Handicap Scale is also very important because then you can relate the muscle strength to the daily life activities of the patient and you see what it really means. And this is also where patients can really contribute to learn more about disease, to learn more about the natural cause, and to learn more about treatment effects. So what we found in untreated adult patients, a decline in mobility, respiratory function, and ability to perform functional activities is already measurable in 2 years' time. I will devote a few slides to the results of the separate questionnaires on daily life, so the Rotterdam Handicap Scale, the Fatigue Severity Scale, and the SF-36. I'll start with health-related quality of life. Of course, this is very difficult to measure. And what we used to measure it on a group level was the SF-36, which is a questionnaire with 36 questions. And you get a score on 8 domains of health-related quality of life. And this is kind of a profile that you get on a group level of a group of patients. And the blue line is a comparison, a healthy population, and the red line are the Pompe patients that participated in the IPA survey. And I'll just pick out a few things. This is the physical functioning and the role functioning physical domain. And here you see that Pompe patients significantly differ from the general population. So on the other hand, this is emotional functioning and mental health. You see that there is no difference with the general population. I'll move on to the Rotterdam Handicap Scale. I just showed you the results of 2-year follow-up. And this slide, I just wanted to show you the distribution of scores in the total study group. And we plotted here the Rotterdam Handicap score versus the number of hours respiratory support. And these patients with dark dots do not use a wheelchair, and these patients, they do. And you see there's a large variation between patients, but also you see that there's a correlation with the respiratory support and with wheelchair use. And what we concluded from our analysis of the results of the Rotterdam Handicap Scale was that Pompe disease has a large impact on the daily life of patients. And I did not show you that, but we found it when we looked at the individual items. In particular, the ability to fulfill their work or study was affected. And you may all know that, of course, but it's very important information to have on a group level because you want to get treatment and you want to get it reimbursed and you have to show the impact of disease on the daily life. And we think that the results from this scale are very helpful to do so. So then I'd like to show you some results of the fatigue scale that was completed. The fatigue scale, fatigue severity scale, is a scale measuring fatigue and its impact on daily life. And you get a score between 1 and 7. and a higher score means more fatigue. And in literature, it's generally agreed that a score above 4 means fatigued, and a score above 5 means severely fatigued. And we made groups. This is a group of patients who are still ambulatory and do not need any ventilation, patients who need a wheelchair, patients who need ventilation, and patients who need both. And you see that fatigue increases slightly with increasing disease, disease progression. But you also see that even the patients who are still ambulatory and do not need any ventilation yet already score very high on the fatigue scale, especially when you compare it to the score of healthy controls, which is 2.9. So what we concluded was that fatigue in late onset Pompe disease is much more prevalent than previously thought, both in mildly and severely affected patients. So if I return to the question I raised at the beginning of my presentation, shouldn't we be asking patients if they are better? I think the IPA survey has shown the value of patient-reported outcome measures. It has shown it leads to a better understanding of the impact of the disease on patients' lives. It indicates the relevance of treatment for late-onset Pompe patients, and that's also something that came up yesterday. So I think in this, the IPA survey has been very important. I've shown that it's complementary to other outcome measures, such as laboratory values, muscle strength, and respiratory function measurements. And if you have both kind of measurements, you get a very complete picture of the impact of the disease on the patient's lives, on the natural course, and we hope in future also on treatment effects. And as the ultimate goal of any treatment is that the patient feels better and is able to resume his or her previous activities, I think it's only logical that we have gone to the patient and asked their point of view. Also, the FDA recently put a document on the internet stressing the importance of patient-reported outcome measures and giving some guidance on how to use them in the design of studies. So to end my presentation, I think structured follow-up will be very important. Also now that Myosin is approved and available to a wider group of patients, because it is necessary to further advance our knowledge on the natural course, to identify prognostic factors, for the indication and timing of enzyme therapy, and to fully evaluate long-term enzyme therapy effects. And I think patient-reported outcome measures are a very large part also to fully evaluate the long-term effects. So this is what I wanted to show you today. I hope I have been able to show you what kind of results we got from the IPA survey, and I hope I have motivated you to keep participating in this project. I want to thank many people from Erasmus, from the International Pompe Association, most of all participants.
Thank you very much.
Thank you.
Okay. We'll take some questions now. Are there any questions from the audience? Yes.
My daughter was diagnosed at 12 and is 29 now. She's thinking about pregnancy, and I'm wondering if you have done any studies on individuals who've gone through a pregnancy.
No, we haven't. Not in the survey. We did not specifically
Is that
ask for
anything planned or going to be looked at, or shouldn't it
Perhaps
be?
it's a good idea, yeah, but we haven't done it so far.
Because she doesn't have any idea on how it might affect her. Or
Okay, so but we haven't.
Yes.
I'd just like to say that in the Pompe connections, the first version, we have an item on getting children. And there, because we know from other neuromuscular diseases that becoming pregnant is really have an impact on your physical ability, like breathing. Because if your stomach is full with a baby, then your diaphragm will be how do you say that? Affected. But there is an issue I can show you when you like. Yeah?
Yes.
The graph you put up, which was the circle with the normal,
Yeah.
and then the Pompe patients, the first line that was going vertical, I think we were about 35% on physical. Yeah, the PF is the physical, is that correct?
Sorry?
The PF line was
Yeah,
for,
that's the physical functioning.
what was the RP? I was just curious what that
It's,
for?
this one you mean?
Correct, yes.
It's the role functioning physical, so how people adapt to, with physical limitations, how they adapt to their normal roles, like being a father, being a worker, being a... Is that clear?
Yeah.
Okay.
Yes, Lena.
I must have missed it. What was the level of residual enzyme activity in those patients who had the same genotype and developed the disease at quite different ages?
It varied between, if I remember well, between 4 and 23%.
But there were differences between
There were
those who developed the disease very early and those who developed
Yes.
it much later.
But go back.
Yes.
There was not really a correlation, because here you see residual activity is higher and they develop the disease more early, and this is the other way around. So it was not really a clear correlation. Is that an answer?
I will take advantage. Were the values done in the same laboratory? Were the values done in the same laboratory, so they are comparable?
They're all done in Arnold's
Arnold's
laboratory.
laboratory.
Yeah.
Okay, okay. And, yes?
I think it's important to know that this is only the subgroup of patients who have the IVS1 mutation, and that overall there is a relationship between clinical severity and presentation of disease, that there is no misunderstanding. It's just a remark. But for the IVS1 group, there is indeed no difference.
I think that's an important point.
That's
Yeah.
a good point to add.
If I may, I'd like to ask a question. When you talk about ventilation, do you in any way, in your questions, identify what kind of ventilation?
Yeah, we did. We made a difference between the between nighttime ventilation and also people who had a tracheostomy. But just for the clarity of the slides, I did not show the differences here.
Does it make a difference whether you just do tracheostomy or you just do nighttime ventilation?
Yeah, there is a difference. There's a,
And can you summarize for us? Okay, sorry.
yeah, I think
Okay.
that's just what anyone would expect. That with increasing time of ventilation, you got increasing handicap and decrease all the other points we measured with regard to disease severity also were related.
But does it change the relationship of respiratory function to being in a wheelchair?
How do you mean?
I think we should maybe discuss
Okay.
this later.
I don't understand the
Okay.
question.
Are there additional questions?
I actually wanted to make a statement back on the issue of getting pregnant. And I say this because all the research says don't, basically. However, I got pregnant and got better. My lung functions, everything seemed to get better with it, and I think it's definitely an individual choice. But I know at the time I got pregnant, I was looking for information out there about what to expect during my pregnancy. So I, again, back up the idea that maybe we should look at a pregnancy study. Because I just met a young lady who pregnancy severely changed hers for the worse as well. So I think it's definitely an individual choice, but talk to a lot of people.
Are there any other questions? Yes.
I was just curious because I'm just learning from being here about the gene mutations, and it did appear that with the adult onset that there was more numbers in a certain kind of gene. Could you explain that a little bit more?
It's only in the Caucasian population that this holds true.
The largest percentage of late onset patients has this IVS1 mutation, which gets some residual activity, and it is in the Caucasian population. It is certainly the largest, the most frequent mutation, but it has also in other ethnic groups. It's also present. So.
I don't think so. I've
It's
never
published
yet.
in this.
With the
it's not
IVS
only
one?
Caucasians, also in other ethnic groups.
Yeah, well, I won't argue, but an admixture, it's primarily, they're
It's primarily Caucasian.
published, there are 160 patients who carry the who have been carried the IVS1 mutation, and they represent about 60 to 70%
Yeah.
Of the late onset patients. And yes. Yes.
I was wondering, it seems like a lot of people have taken many years in order to get diagnosed, and with symptoms starting at varying different times. When you do a study like this, how can you control for that so that you know you're really
Yeah.
comparing like to like, and if you could control it better, or would that maybe give us some of the answers?
Now, in fact, what we did was that we also asked patients when their first complaints started, and we calculated 2 disease durations, one with first complaints as the starting point and one with age-at diagnosis as the starting point. And there are differences, of course, because of what you say. Some people take quite a long time to get diagnosed. But in the end, on a group level, we found out that it didn't make much difference for the conclusions that we drew. So for clarity, we just chose to show you one, and that was measuring from the time of diagnosis. So, is that an answer?
Well, I think that you're going to have to answer
Okay,
questions
fine.
afterwards. And thank you for really magnificent and very, very important
Thank
study.
you.
I think I'm going to turn this over to Dr. Howell, and I'm going to ask someone else to put this plug back in.
I think that Dr. Hagemans' talk was an outstanding entree into the discussion of research in Pompe disease. I think it's very clear to everybody in the audience the extraordinary variability that's seen in Pompe disease, and that variability extends not only to the time of presentation, the clinical presentation, but also the response to therapy. It's an exciting time, obviously. There's a new approved therapy, and et cetera, out there. It's very clear that for babies born with Pompe disease, it's important to be treated very early, which leads us into the issue of newborn screening. And I think that that as we proceed in the research area this morning, obviously, I think it's very clear that newborn screening will soon be a reality. There are currently funded pilot projects, one in Washington State at the current time, and I think you can see immediately some of the complexities that will arise when you get a newborn blood spot that has an extremely low enzyme activity, and deciding exactly what course you should follow, etc. We have an extraordinarily distinguished panel this morning to talk about research areas in the Pompe disease. And as you review the credentials of our speakers this morning, they began their careers and their education in Moscow, New Hampshire, London, and Kentucky. So it's a very diverse group as far as their scientific activity. And we're going to lead off this morning with Dr. Nina Raben, who is a name familiar in the area of research in the area of Pompe disease. Dr. Raben is a staff scientist at the National Institutes of Health at NIAMS, that's the arthritis and musculoskeletal and skin diseases. She has published extensively over many years in the metabolic myopathy. She's published a great deal of information on phosphocorticokinase in addition to Pompe disease, and she's a leading worker in the area of mutational analysis. And she's going to spend time this morning with us talking about the role of autography in the area of Pompe disease. And we will, her official title is this morning, Muscle Pathology and Mechanism of Muscle Wasting, and it's great to have Nina with us, and she's revving up her PC. She brought her private PC. She's from the NIH, you know, and they like to keep things on the up and up at the NIH. I think, let me comment, some of you may not be completely familiar with the National Institutes of Health, which is located in Bethesda, Maryland, which is where I spend my time these days. But it's the Federal Research Institute. It's the largest research institute in the world. And if you have an opportunity to be in the metropolitan Washington area, I'd encourage you sometime to come by. There's 17,500 people there working hard to solve various and sundry problems.
I'm going to talk today about the autophagy, which literally means self-eating. I'm going to talk about the role of autophagy in the pathogenesis of Pompe disease and about its potential role in the outcome of therapy. We all know that the basic concept of enzyme replacement therapy is receptor-mediated endocytosis of the recombinant enzyme. The recombinant enzyme binds to mannose 6-phosphate receptor on the cell surface, and it enters the endocytic pathway bound to the receptor. The receptor enzyme complex moves to early then late endosomes. Here at the acidic pH of late endosome, the receptor enzyme complex dissociates. The receptor moves back for the next round of sorting, and the enzyme moves to the lysosomes where it should degrade the accumulated glycogen. So this is a theory, but in real life, in both clinical trials and in our preclinical studies with animal models, the recombinant human GAA produced remarkable success in terms of reversal pathology in cardiac muscle, but the success in skeletal muscle is less obvious. I'm not going to go through this slide. You've heard beautiful talk of Priya Krishnani and Hans van der Ploeg yesterday about clinical trials, but it is very clear that the reversal of pathology in skeletal muscle is not tremendously successful. In mice, we have quickly found that only type 2 muscle fibers resisted to therapy, as shown here with the light microscopy before and after therapy, and those purple dots represent glycogen. As you can see, in this particular case, there's basically no glycogen clearance after enzyme replacement therapy. After months on therapy, what we found was that this type 2, that the reduction of glycogen in type 2 fibers was very modest. What was interesting about this study was that in untreated animals, shown here in blue, those therapy-resistant fibers accumulated much less glycogen than cardiac muscle or type 1 muscle fibers, which cleared glycogen very beautifully. So the first message from this study was that, quite paradoxically, the amount of storage material is not the major factor which determines the effectiveness of enzyme replacement therapy. So these studies led us to explore the differences between the fiber types in an attempt to understand what is it that makes type 2 fibers resistant to therapy. Here are shown some very well-known differences between the fiber types, and I'm not going to go through that. In addition, what we found was that there is a quite different distribution of the lysosomes in type 1 and type 2 fibers. We also found the low level of several trafficking protein, as shown here, which are important for the delivery of the recombinant enzyme to the lysosomes, and in particular, relatively low level of mannose 6-phosphate receptor. You remember this is the receptor which delivers the enzyme to the lysosome in type 2 fibers compared to type 1 fibers. So these were intrinsic properties of type 2 fibers. And we found these differences both in wild type and in the knockout. And so this could obviously contribute to their differential response to therapy. But the elephant in the room was the presence of huge autophagic areas in type 2 fibers. In type 1 fiber, one can see enlarged lysosomes filled with glycogen, as is typical for Pompe disease, but only in type 2 fibers we also see this huge area of autophagic activity. Although noted many years before, the presence of autophagy had largely been ignored, and it is actually surprising that the autophagy was not directly linked to the pathogenesis of the disease, because after all, autophagy is a lysosomal degradation pathway. It is a normal process of degradation and recycling of long-lived proteins and damaged organelles. The process starts with the formation of a membrane which engulfs part of the cytoplasm and damaged organelles, resulting in the formation of double membrane structure called autophagosome. Autophagosome fuses with lysosomes where the membrane and the content is degraded and the product of the digestion are recycled back into the cytoplasm. Autophagy is a critical survival mechanism under nutrient deprivation. LC3, a highly specific autophagosomal marker, is present on the membrane throughout its elongation process, and it disappears when the autophagosome fuses with the lysosomes. So the lysosome is the end point for both the endocytic pathway, the route by which the recombinant enzyme goes to the lysosome, and the autophagic pathway. What is important here is that autophagosome can also fuse with late endosome or even with early endosome, resulting in the formation of intermediate structure called amphisome, and this can result in mistargeting of the recombinant enzyme. Under normal condition of productive autophagy, autophagosomes or amphisomes are quickly degraded by the lysosomes, and this LC3 positive structure, double membrane autophagosomes, are barely detectable. Electron microscopy established the presence of autophagy in type 2 fibers, but when we began to isolate single muscle fibers to study the geographic distribution of the structures which were seen by electron microscopy, we realized that autophagic areas were huge, that they extended almost the full length of the fiber, and strikingly, they were often located in the center of the fiber, as shown here with this specific autophagosomal marker, or with another marker, LAMP1 or 2, which is a specific marker for late endosomes and lysosomes. Once recognized, the autophagic areas could be clearly seen in fixed or live muscle fibers without any staining by low-resolution transmitted microscope, as shown here in the knockout fibers. In the previous slides, you have just seen the autophagy in a single muscle fiber. Here, we can observe autophagy in multiple fibers by taking advantage of a technique called second harmonic generation imaging. I'm not going into details of this technique, but what's important here that the method allowed us to see the autophagic buildup in an intact muscle biopsy without any staining. And in a series of images scanning through the type 2 knockout muscle, you can see huge black holes within every fiber. Those holes are filled with autofluorescent material, which is shown here in green. Here is a three-dimensional image processed from a type 2 knockout fiber, showing the volume occupied by this autophagic buildup within the fiber. And the question is, what is inside those autophagic bags? A closer look of the LC3 and LAMP-positive structures in the autophagic area shows that the two markers often co-localize. In this slide, the late endosomes and lysosomes are stained in red, and the autophagosomes are stained in green, the same here. So the presence of double positive structure, shown in yellow here and here, reflects either excessive accumulation of amphisome or the inability of the lysosome to digest the contents of the autophagosomes, or both. The process starts very early. Excessive autophagy can be seen in 1-month-old knockout mice well before the symptomatic disease, as shown here by the presence of isolated vesicles clearly stained with autophagosomal marker LC3. Interestingly, at this early stage, it is not uncommon to find small damaged lysosomes inside the autophagosomes, as shown here. So in an unusual role reversal, it appears that the autophagosomes engulf the lysosome rather than reverse. As the disease progresses, the autophagic buildup appears to interrupt muscle striation, as shown here with the myosin staining in red. And even in very young animals, one can see massive accumulation of lipofuscin, a presumed product of oxidative damage, as shown here for a 6-month-old knockout mice with Hurt staining. These so-called, not pejoratively, but there is a biological term, the biological garbage. So these biological garbage filled bags grow with age, and they seem to have a greater effect on the muscle architecture than the expanded lysosome seen here at dark spots outside the autophagic area. And the process is clearly disease-related because the autophagy is not detectable, as you can see, in age-matched wild-type animals. At a later stage, the illness is the later stage of illness, the integrity of the vesicles in these autophagic areas appears totally lost, and only remnants of vacuolar membranes can be visualized. And the question is, what do these findings have to do with the problems we set out to solve, the failure of recombinant enzyme to clear skeletal muscle of glycogen? To address the issue, we looked at the endocytosis and trafficking of the labeled recombinant enzyme in live type 2 fibers. In wild type fibers, the endocytosed enzyme goes to lysosomes, as shown here. We know that these are the lysosomes because it's a very typical pattern of staining we see with a specific staining for the lysosomes. Strikingly, in the autophagic area, which can be clearly seen in this phase image, becomes a sink of the recombinant enzyme. Now, and the question is, what is the relevance of these studies to what we see in humans? And I want to emphasize here that this distinction between the type 1 and type 2 fibers was an opportunity for us to see and try to identify what makes one fiber types more resistant to therapy than the others. So what is the question about the humans is not what is the predominance of type 1 or type 2 fibers affected, and whether type 2 or type 1 fibers respond equally to therapy. The question for us in human study was only one: whether autophagy plays as important role in pathogenesis of the disease as it is in knockout mice. And what we found was that in humans we see many of the same things which were observed in mice. Autophagy seems to be a big player in the pathogenesis of the disease. First, one can clearly visualize autophagy in muscle fibers from patients with Pompe disease. And just as we were able to follow the age-related progression of the disease in mice, we can follow the stages of disease progression in humans by analyzing multiple fibers from a single patient, because the phenotype of muscle cells, at least in adult patients, ranges from normal, as shown here, to mildly affected, to most severely affected, with everything in between. So we can try to deconstruct the disease into a series of stages of disease progression. And what is remarkable is that these stages are, again, very similar to what we see in mice. At the earlier stages, one can see a formation of a series in the core of the fiber of a row of relatively small lysosomes, which are already unable to digest the content of the autophagosomes. So these are autophagosomes at a very early stage of the disease, and this is something you never see in normal skeletal muscle. The process occurs at multiple spots along the fiber, as shown here with lamp staining, which is specific, again, for lysosomes. At a later stage, the area expands, and autophagosomes, shown here in green, are clearly seen in the autophagic area in the core of the fiber. Furthermore, similar to what we observed in mice, the lysosomes can be found inside the lysosome, inside the autophagosomes, rather than the other way around. As you can see, the red is clearly inside the LC3 positive structures. There is no question that the expanded autophagic area and the autophagic buildup, hope the movie is playing, yes, and the autophagic buildup constitute a significant part of pathology in humans. But what is remarkable that it seems that this buildup constitutes the major pathology in the affected fibers. Because in fact, we do not see largely expanded lysosome outside the autophagic area. And I will try to illustrate it in the next couple of slides. In some fibers, the lysosome outside the autophagic area are very small, and as you can see, they do not seem to cause any muscle destruction. In other fibers, the lysosome outside the autophagic area are clearly enlarged. They are shown here in red, but they too do not seem to be very harmful. Here is another example showing that the autophagic buildup constitutes the major pathology in the fiber. Finally, in the most severely affected fibers, the vacuolar structure is totally lost, just like what we observed in older mice. What exactly precedes this stage of the disease is very hard to define. It may be that at a certain point, the disease progresses very rapidly. And at this later stage, the therapeutic enzyme, for obvious reasons, cannot do too much good. First, because the muscle structure is almost totally lost, and also because remarkably, the whole area is totally devoid of mannose-6-phosphate receptor, the receptor which brings the enzyme to the lysosomes. I've just shown you the heterogeneity among the fibers, but there is also a tremendous heterogeneity even within a single fiber. This is a composite of several images taken along the length of a single fiber. The data from both our mouse models and human studies led us to reconsider the view of the pathogenesis of the disease and the mechanism of skeletal muscle damage. The current view, which was put forward more than 20 years ago, is that muscle damage occurs because, unlike in other cells, the lysosomes in muscle cells have very limited space in which to expand. And this results in mechanical pressure and rupture. We're not arguing with the idea of vesicular rupture, and in fact, at later stages, we do see the disintegration of the vesicular membrane. However, the stages leading to this final stage are clearly at odds with our experimental evidence, both in animals and in humans. The data strongly indicate that it is not the global expansion of the lysosomes which causes skeletal muscle damage, but rather some yet unknown abnormalities in a subset of lysosomes which do not allow them to recycle autophagosomes and their content. So autophagy, which is known to be induced by starvation, sets up the condition for disruptive buildup and diversion of recombinant enzyme away from lysosome. In conclusion, we think that enzyme replacement therapy addresses the problem of accumulation of glycogen in swollen, expanded lysosomes, and it addresses it very successfully. But we think that it is critical at this time to look beyond the lysosomes, because it is increasingly evident that other processes, and in particular autophagy, affects the outcome of therapy. Thank you very much, and I would like to acknowledge many people who contributed to this, and most of all, my boss and the person who I work with, Dr. Paul Plott, who sends his warmest regards to the House's family and to everybody. Thank you very much.
We're going to move now to Dr. Robert Mattaliano, and Bob is the Group Vice President for Protein Development at the Enzyme Corporation, and he is going to, he is this morning going to talk to us about the development of protein, of clinical grade recombinant proteins to support the clinical phase development, and he's now going to talk to us about the quest for other contributing factors.
When I think about Pompe disease, I think about one thing, and that's variability. You've heard it very elegantly from a number of speakers that there's variability within age of onset, there's variability in the main tissue types involved, and also the degree to which those tissues are involved. And in large part, that variability correlates at a global view with the amount of residual activity seen in the muscle cells and generally experimentally examined in fibroblasts. It's the genetic mutations or the genotype of the individual who is affected that really drive what that measurable activity is. And typically in the late onset population, for example, we see measurable activities of up to 40% of normal, whereas in the infantile population we see activities of often less than 1 or 2% and sometimes zero in the so-called CRIM-negative patients. When we think about enzyme replacement therapy, we also have to consider the degree of pathological damage which exists when the treatment begins. These are light microscopic images of some quadriceps muscle in two cases that we have experience with, case A and case B, and the purple stain is the glycogen showing the very different pathology, and Nina's pointing, showing the autophagic buildup probably in this bottom panel, case B, pre-ERT. But certainly in case A, upon treatment for 1 year, we clear most of the glycogen. You see the muscle fibers returning to a near normal histological state. But that's not the case in case B. So we're very curious to know why that is. Why can't we get there? Certainly some models suggest that it is the lysosomal rupture and the formation of extralysosomal glycogen where the ERT is not going to work because the pH is wrong is one of the contributing factors. But Dr. Raben has very elegantly described another major factor that is now, I think, the 300-pound elephant on the table, and that is the role of autophagic buildup. This is an example from one of her papers showing the type 2 fiber in the mirroring model. Again, here's the autophagic area disrupting the microtubular structure of the muscle fiber. So there are a number of factors that we often think about in the context of why skeletal muscle may be refractory to ERT. And why is the dose so high relative to other enzyme replacement therapies? The dose is almost 10 to 20 times higher than what we use, for example, for Gaucher or Fabry disease. Contributing factors include overall muscle mass. As Dr. Van der Ploeg pointed out yesterday, it's up to 40% of the mass in an adult. It could be the degree, and probably is very likely degree of pre-existing pathology. There's certainly a point of irreversible damage. It could be altered function of the autophagic or endocytic pathways, again with a likely point of irreversible damage. It could be differential rates of glycogen accumulation between muscle groups and fiber types. It could be immune response to the enzyme replacement therapy. We heard yesterday that those patients who are CRIM negative in general don't have a good outcome. We'll talk more today about patient genotype and genetic background. And it could also be inefficient GAA targeting. In other words, the GAA could have inappropriate post-translational modifications or traffic to non-productive receptor sinks. And my colleague Cheng, after I speak, is going to give a presentation on some things we're trying to do to improve GAA targeting in a second-generation molecule. So, what are we looking at? Pathophysiology is still an important area of investigation, and there are a number of factors that we're still examining histopathologically. They include severity of the disease at baseline. We can look at glycogen load and other ultrastructural damage, such as autophagic buildup. We're also interested in looking at fiber type in humans and the proportions of slow versus fast switch fibers, because as you just heard, the ability of enzyme replacement therapy to clear glycogen from those fiber types appears to be different. We're also interested in the ability of the enzyme to get to the affected tissue, which in part may relate to capillary density. So the question is, how do these parameters correlate with glycogen clearance upon ERT? And we've done a number of studies looking at several things. First of all, we looked at satellite cell activation shown here in these fibers using a nucleomyogenin stain, which is shown in this green dot here. We looked at fiber type using the HTPA stain. Slow twitch fibers don't stain, whereas the fast twitch fibers stain dark. We've also looked at capillary density using immunohistochemistry, an antibody against CD31, which stains endothelial cells. And the overview of our observations is that persistent satellite cell activation is observed in muscles with very severe pathology and poor glycogen clearance. It's as if the muscles are trying to repair themselves but are unable to do so. There appears to be a slight correlation between glycogen clearance and a high proportion of type 1 fibers. And that's consistent with the story you just heard, whereas the type 2 fibers are more likely to have autophagic buildup and are less likely to be cleared of the accumulated autophagic vesicles or glycogen. And also with regards to capillaries, at least in quadriceps, we don't see a correlation with glycogen clearance so far. So I'm going to turn my attention to some more studies on patient genotype and genetic background, some areas that we're trying to mine with regards to looking for these other needle-in-the-haystack factors that might affect clinical outcome. The GAA gene, as you know, it's a very large gene. It's almost 3.5 kilobytes in size. There are 200 published disease-related mutations, as you just heard earlier this morning. And again, I call your attention to a very good website maintained by Arnold Roycer and his colleagues at pompacena.nl, which lists all of these mutations and the correlations with phenotype. However, in our studies, we've also looked for a lot of mutations, and we've seen the major mutations. This is the IBS mutation that Marluz talked about. This is allele frequency, not patient frequency. And what you see here is that the major genetic mutations occur in our studies, in our clinical studies, at about the same rate as or frequencies as those that are in the literature. But one of the hallmarks of Pompe disease is that there are numerous private or rare mutations. And that's one of the challenges for our studies, that unlike Fabry or Gaucher disease, where a handful of mutations often make up 60 to 70% of the total number of mutations, that is not the case for Pompe mutations. In fact, new mutations are being observed all the time. In fact, we've identified, in collaboration with the group in Rotterdam, 100 new mutations, which will soon be added to the mutation database, bringing the number of mutations to over 300 today and growing. The observation is that GAA mutations don't explain disease severity alone. And there's some examples as to why that is. And as you heard this morning, one of them is the IBS mutation, which is a leaky splice site mutation and results in decreased levels of transcription of the normal DNA. When coupled with a nonsense mutation, this one, TYR609 stop, it's been associated with an adult onset phenotype. However, when coupled with another nonsense mutation, it's coupled with a juvenile onset. So this says, at least between those two individuals, there's likely differences in the transcriptional machinery. Another example is the ARG600 cis mutation, which has been shown to have zero activity in fibroblasts, but interestingly has a low level of residual activity in muscle. When paired with a null mutation, we see a juvenile form of the disease. However, when this mutation is homozygous, we actually see a more severe infantile form of the disease. So these kinds of data taken together suggest that mutations alone don't explain disease severity. This is a table similar to the one that Marlowe showed this morning, and I'd just like to point out that we looked at in our clinical studies of late onset patients a cohort of individuals who have the exact same mutation, and yet their residual activity levels range from 11 or 12 to 34%, almost a factor of threefold. So clearly there's other factors involved. There's been a recent publication by the group in Italy which has shown that a similar IVS mutation paired with this TRP746 stop, in 9 patients they've shown between 0 and 18% residual activity, again showing this diversity of residual enzyme activity in patients with the same phenotype. So the question or the issue is that even with the same genotypes, individuals have different residual activities, again suggesting that some other factor or factors are in play. So what can we do? We catalog the small stuff, we look carefully at single nucleotide polymorphisms, we build haplotypes, and we compile the clinical course of the data for each person. At Genzyme, because we're doing a lot of genotyping and sequencing most of the genomes for our patients, we track 73 SNPs, which are accessible readily, that are in exons or close to intron-exon junctions. And these SNPs can be ordered into haplotypes for the alleles in individuals. And we do know that there's racial and or ethnic-specific variation. We built haplotypes. This is a similar table to the one that you saw earlier. And in our clinical experience, we've actually seen the IVS1 mutation on 9 different haplotype backgrounds. Background variation for other common mutations is evident. So the question is, to what extent does this variation contribute to or account for the observed differences between individuals with regards to clinical progression of this disease? Now, we spend a lot of time looking at the GAA gene, but we shouldn't just look there. It's the old, if you only look at we're only looking under the street lamp for your keys, you may not find them. So we're looking in other places. And the question is, are there accessory or modifier genes which might be involved that result in a differential clinical response? So everybody has their favorite gene set that we look for, and it's called the candidate gene approach. For example, there's a number of genes in the disease pathway that may be potentially involved in the disease pathway. We think about genes associated with fiber type determination, for example, the ACE gene or the actinin-3 gene. We're also interested in mutations in the mannose-6-phosphate receptor family and in the LAMP family because they may be contributing factors. But this is a very, very time-consuming process to do these individually. So we've taken a slightly different approach, and that is to do differential RNA expression analysis from samples from patients who have, from which samples were obtained at baseline and after 1 year of enzyme replacement therapy. Before we did that, we created categories for how patients have responded. So patients were sorted into classes based on the 52-week clinical and biopsy results from the 1602 trial, which is that study in infants you heard about yesterday that are less than 6 months of age. And we defined them quite rigorously into 3 cohorts. Motor responders, these were infants who had clinical improvement and a decrease in quadriceps glycogen content by both histological and biochemical methods. 6 out of the 18 patients fell into this criteria. On the other end of the spectrum, because we're trying to measure big differences between the spectrum with the poor motor responders, those who had clinical decline and also in which we observed an increase in quadriceps glycogen by either histological or biochemical methods, and this was 3 of the 18 patients. The rest of the patients fell into some intermediate group. So what we did was to collect quadriceps biopsies at time 0 and after 52 weeks of enzyme replacement therapy, extracted RNA from these samples, hybridized them to a chip containing 47,000 of the human genome transcripts to see which messages were being expressed, analyzed the data, and then analyzed that information in 2 different ways. We took an unsupervised approach, that is to ask the computer who is different versus who is alike. And we also took a supervised approach, which is to tell the computer who is alike and look for similarities. And then we confirm those results by quantitative PCR. And I'm going to show a couple of examples in just a moment. So this is a heat map. This is what the gene expression profile analysis looks like for motor responders versus poor responders. Motor responders are in blue, poor responders are in red, and the intermediate responders are in purple. And what you may be able to ascertain is that really it follows the motor responders cluster into one branch of this tree, if you like, whereas the other individuals, the motor responders and the intermediate responders, cluster into other branches. And this says that the poor motor responders, their expression levels, their gene expression levels, are more closely related to one another than to those individuals who have a good response or an intermediate response. You can also apply a lot of fancy bioinformatics tools to this problem, and I'm not going to go through all of these. And this is the supervised approach. Use these tools to look for expression of signals that are statistically significant. And when we do that, we come up with lists of genes that fit statistical criteria to assert that their expression is in fact relevant and different between the responder and the non-responder groups. And I'm not going to go through this list of genes. I'm just going to show you some examples as I finish up here briefly. So here's one called CD79A. I'm going to tell you what this is in just a second. But here's the relative fold expression in poor responders. It's between 20 and 100-fold greater. This is the actual qPCR data versus the motor responder. So this is one gene where there appears to be a difference in response. CD79A is a pan-B cell marker, and it's physically linked to the surface of B cells, to membrane immunoglobulin forming part of the B cell cell antigen receptor complex. And it has it's shown here on this IgA right here. And it's a heterodimeric molecule which is comprised of two polypeptide chains, CD79A and CD79B. However, they're not expressed in concert. They can be differentially expressed. And we're still trying to understand just what this means. One more example is myosin heavy chain 1. Now, this is a protein which is upregulated in motor responders versus poor responders. And so protein is interesting because it's more abundant in type 2B fibers or the fast-twitch muscle fibers. So it makes us wonder if there's some sort of variation in the cellular architecture of those individuals who respond and have a different pathophysiology in their type 2 fibers, for example, relative to the poor motor responders. So where are we now? So using this unsupervised hierarchical clustering approach, we've seen three major branches. The groupings are consistent with the clinical findings. And what we're trying to do is to see whether baseline gene expression patterns can be used to predict patient response to treatment. It's very early days, and I've only presented a couple of examples that give us a little bit of a glimpse into what that possibility might be. We've also looked at the comparison of gene expression patterns between groups, and we've identified multiple differentially expressed genes. I've just shown you a couple of examples, and the functional significance of those genes is still being evaluated. But the bottom line right now is that beyond genotype, in infants, CRIM status, and the degree of pre-existing cellular damage, looking for prognostic factors to differentiate motor responders versus poor motor responders remains an elusive and challenging problem. The future, I think, in part, is to begin to look at families, and we have a collaboration with an investigator in Brazil, Dr. Sully Marie, who has a number of families with affected siblings. And so we've begun a similar process to look at the differential RNA expression in members of these families. All of the family members in several cases have agreed to provide biopsy samples, biceps and triceps, and allow us to examine the differential expression of these genes. And this case is a particularly interesting case because here's a brother and sister who have the exact same genotype, and yet the onset of their disease was very different. Her age of onset was 28 years old. You can see the glycogen buildup shown here. They have the same degree of residual activity, and yet his age of onset was 42 years of age. So we'd very much like to understand what these other genetic factors are which drive these differentials, and we're still trying to figure that out. So I'm going to stop, acknowledge a number of collaborators. I'd also like to thank the group in Rotterdam that's helped with some of the fibroblast expression and haplotyping work, and thank you for your attention.
It was a very good talk, and we're going to continue along the new research development area, and we will next hear from Dr. Cheng, who is Group Vice President of Genetic Disease Sciences at the Enzyme Corporation, and he's actually responsible broadly for discovery and preclinical research, etc. And he's going to talk to us about the new generation enzyme and gene therapeutics for Pompe disease.
So, good morning. I'd like to thank the organizers, in particular the Howell family, for their kind invitation and opportunity to attend this meeting and to present our work here this morning. And this has really been a very informative and very educational for me. So as Dr. Howell indicated, what I'd like to do for the next 20 minutes or so is to tell you about our ongoing efforts at trying to evaluate and develop what we believe could be improved therapies that we could bring to bear for use in the treatment of this disease. And as you think about different approaches that one can use, we can think of different modalities, and that includes not only enzyme replacement therapy that you're all very familiar with, but also what I would refer to as emerging therapies, such as gene therapy, or indeed even cell-based therapies, or even small molecule drugs, as illustrated here. But for this morning, what I'm going to do is just focus my presentation on our efforts at trying to look at ways by which we can improve upon the properties of the enzyme that we have today, and tell you a little bit about the emerging therapy that I'm going to refer to as gene therapy, that you're aware of as well. And in thinking about ways by which we can improve or develop what we refer to as next generation therapies, one obviously needs to think about what are the challenges that are associated with the current therapy. And you really heard a lot about this from the different speakers yesterday and this morning. So for example, as illustrated here, we know that the primary organs that are affected in Pompe disease, such as the cardiac and skeletal muscles, these tissues are not easily accessed, as you heard from Dr. Are not easily accessed by, okay, I'm going to move this up a little bit higher. Okay, is it better? Yeah. Okay, sorry about that. So these tissues apparently are not easily accessible by venous infusions of the enzyme. Indeed, we know that a large proportion of these enzymes are actually sequestered, as you heard earlier, by what we refer to as non-productive organs, such as the liver, which is not significantly affected in Pompe disease, and that a relatively small amounts of these enzymes are actually delivered to the relevant tissues, which are the skeletal muscles, which is what we want to hit. And indeed, this is borne out from the clinical studies that you heard from both Dr. Krishani and Dr. van der Ploeg, and that suggests that relatively high levels of these enzymes, he's trying to play with the microphone's volume. Or maybe this. Okay, is that better? Okay, that's good. Is this better? Okay, I'm going to stay still here then. So these problems that we illustrated here might account for the reasons why we need relatively large doses of these enzymes for clinical efficacy in the range of around 20 to 40 milligrams. And these are much higher than what we typically use for the other lysosomal storage disease for which we develop enzymes, which typically range in 0.5 to 2 milligrams per kilogram. And that associated with the introduction of these recombinant enzymes, as you heard, this elicits the generation of antibodies, which Dr. Krishani suggested may impact clinical efficacy. So to address some of these issues, what we've been working on for the last couple of years are two approaches. And the first is what we refer to as carbohydrate engineering. And what we're trying to do here is to change the properties of the enzyme so they are better targeted to the tissues that are receiving less than optimal amounts of these enzymes. And this is not a very novel concept, because this is what we use successfully for a different lysosomal enzyme called Cerezyme, which is the enzyme that we use to treat Gaucher disease. I'm going to tell you again about another approach, and that would be gene therapy. And the reason why we consider this is because gene therapy offers the possibility of continuous production of this enzyme in the body of the patient. And we will argue that this mode of production and delivery, the continuous bathing of the affected organs, might result in better clearance. And I'm going to start off by showing you the progress we made in our efforts to improve the delivery of the enzyme into the affected tissues. Now, you already heard from Dr. Raben this morning that the primary receptor that is involved in the recognition and uptake of this enzyme is a so-called mannose-6-phosphate receptor. Now, we know that myosin, the enzyme that we currently have, only contains a modest amount of the important marker the mono-6-phosphate-bearing oligosaccharides that are involved in binding this receptor and that's involved in the uptake of this enzyme. So based on this observation, we rationalized that what if we increase the number of these important recognition markers on the enzyme, will this now improve its attractiveness or affinity for this receptor and in consequence increase the delivery of these enzymes to those tissues? So this is a scheme that we use to affect this change. So what we did was to actually synthesize a synthetic sugar moiety that contains the important mono-6-phosphate residues. And what we did was to chemically conjugate this synthetic glycan onto the sugars on the existing enzyme that we have today. And the resultant molecule that we generate, I'm going to refer to as NeoGAA. And indeed, if we're successful in our endeavors, this would be the molecules that we generate. So this is the current enzyme myosin. This is the protein component represented here with the 7 oligosaccharide side chains and showing that it does indeed contain the mono-6-phosphate, the residues that are important for recognition by the receptor. And if we are successful with the conjugation that I mentioned, what we would generate in the form of NeoGAA would be a molecule that contains a lot more of these mono-6-phosphate residues. So we obviously wanted to ask whether this chemistry that we use affected the activity of the enzyme. And indeed, when we looked carefully at the modified enzyme, shown here as NeoGAA, and we compared it to the unmodified enzyme, we showed that we did affect the enzymatic activity as illustrated by a change in the so-called KMs or the Vmax. This measure the activity of the enzyme. So with the chemistry that we use, we did affect its activity towards the negative end, so it's less active. And we also showed that this modification resulted in a somewhat altered half-life in the systemic circulation. But as expected, we showed that with the chemistry that we used, we did increase the number of these important mono-6-phosphate residues. We also showed that with the chemistry that we used, we also altered some of the amino acids. But despite these changes, we did show that by the addition of these mono-6-phosphates, we did successfully increase its affinity for the receptor and consequently greater uptake of this enzyme by muscle cells. So what is shown here on the right is a tissue culture experiment. So what we did was add increasing amounts of enzyme, either the unmodified enzyme shown here or the NeoGAA, and ask how much of the enzyme is taken up by the cells. And as you can see here with the unmodified enzyme, as you add more enzyme to the cells, as expected, more is taken up, as illustrated here. But I hope you can appreciate that with the NeoGAA, we can demonstrate that this enzyme is taken up much faster and in greater quantities. But then they said, well, this is all very nice in a tissue culture cell. Well, what does it mean in an animal? So what we've done is to ask whether this improvement or modification we made can translate to a change in its performance in a mouse model of Pompe disease. And this is a mouse model that Anina had generated. So what I show here is a resultant experiment where we introduce either the unmodified enzyme or the Neo enzyme into the mice. And these mice were given 4 weekly infusions of either the unmodified or Neo. And then we assayed for the levels of glycogen in the heart or in the quadriceps as illustrated here. And if you look at the yellow bars, these are the levels of glycogen in the untreated mice, so very high levels of glycogen as you might expect. And if you treat this mice with the unmodified enzyme shown here in red, we saw, as one might expect, a dose-dependent reduction in glycogen. So the more enzyme we use, the greater the glycogen reduction that we saw. And that's what you expect. And if we did the same for the NeoGAA, which is shown here in the light orange, we saw exactly the same thing. If you use more enzyme, you saw greater clearance of glycogen. But what we showed, in addition, it's very nice, is that we can now demonstrate that we can get a similar extent of correction with the unmodified or with the modified enzyme. But with the modified enzyme, we can do this with a much lower dose of the enzyme. So with Neo, we can see the same change using a 5 to 10-fold lower dose of this modified enzyme. And in the muscles, we can show that we can get, again, a similar level of correction using approximately 5-fold lower dose. So with this modification that we've done, we can show that we can increase the amount of enzymes that's delivered to the skeletal muscles, as illustrated here. And we can show this effect not only biochemically, but we can also visualize it when we looked at the histologic sections from these mice. So again, you're now very familiar with this kind of presentations. So what I'm showing you here are the sections in the heart and the cordyceps of mice that were untreated. And if we treat these animals 4 times with the unmodified enzyme, we saw, as expected, a reduction in glycogen, more so in the heart than the cordyceps, and that's the basis for the use of Myosin that we have today. But I hope you can appreciate that with the neo-GAA, which is the modified enzyme, we saw even greater clearance of glycogen in both the heart and in the cordyceps. So you can say, well, that's all very nice that we can clear more glycogen, but what does that mean? So what we've done is to ask what this means in terms of motor function and coordination in the mice. And we can do this by using what we refer to as a rotorod test. So what this actually involves is you place the mouse on a rod and you spin it at increasing speeds, and you measure the ability of the mouse to stay on this rod. And in the so-called rocking rotorod mode, shown here, what we do here is we actually stop the spinning and actually reverse the direction of the spin, and we ask the mouse to turn around and run in the opposite direction. So obviously, mice that are unaffected by diseases, or wild-type mice shown here in yellow, they perform very well on this test, and they stay on this rod for up to 50 to 55 seconds, which is the longest extent we allow the animals to perform on this assay. In contrast, as shown here in red, this is the untreated Pompe mice, and even at 4 to 5 months old, when we first started testing these animals, they already show a deficit, so they fall off much faster, which is what you might predict. And as the animals age over the period of 6 months, we see a progressive loss in function, as indicated by yet a shorter time that they're able to stay on this test. But very gratifyingly, you can treat these animals with the enzyme replacement therapy, weekly infusions for a period of 6 months. We're able to demonstrate that we can rescue this motor function and coordination is illustrated by now an improvement in the times that they can spend on this test. And more importantly, what I show you here is that with the neo-GAA, we can get a level of performance that is equivalent to that of the unmodified enzyme. But that what you should note here is that with the neo-GAA, we're using 1/5 the dose that we use here for the unmodified enzyme. So the greater clearance of glycogen that we're able to realize with the neo-GAA translates to improved performance when we measure using this assay. You can also measure muscle strength, and this is a very simple assay where you put the mouse on a wire grid and then you invert it and you test and measure the length of time the mouse is able to hang on to this grid. And as you can see here in wild-type mice, they again stay on for around 55 to 60 seconds, again the longest time we allow these animals to do this. And in the red is the untreated Pompe mice. And again, as you might expect, as the animals age and there's progressive accumulation of glycogen, you see a progressive loss of muscle strength. And again, you can demonstrate if you treat these animals with a neo-GAA, you can demonstrate a restoration, a lack of loss of muscle strength in these animals. And that this, again, is better than that of the unmodified enzyme. So I hope, in summary, for the first part of my presentation, you agree that this approach of chemically conjugating a mono-6-phosphate residues under the resident enzyme can result in improved delivery of the enzyme to the affected muscles, and that this translates to greater clearance of glycogen from all the muscles, and that we can get a comparable reduction in tissue glycogen in the muscles using a significantly lower dose of the modified enzyme, and that this translates to improved motor coordination and muscle strength, leading us to propose that in this approach of carbohydrate engineering to improve the affinity of the enzyme for this receptor may indeed represent a way by which we can improve the therapy that we can provide to patients with Pompe disease. However, going forward, we clearly need to further optimize the processes that we can use to affect this change. So I'd like to do now is to tell you about yet another approach that we're looking at, and this is the approach of gene therapy that I mentioned earlier. And the reason why we've been looking at this particular technology is because we believe it offers some advantages over enzyme therapy. So for example, for one, because gene therapy offers a possibility of sustained production for a very, very long period of time following one administration, I hope we can all agree that if successful, should translate to an improvement in the quality of care that we can provide to patients that currently have to undergo biweekly infusions of enzyme. Because the enzyme is produced continuously in your body rather than administered intermittently by enzyme replacement therapy, we would also argue that that might provide better delivery of the enzyme to the organs that are not well served by the periodic administrations of protein. And yet another virtue, and I'll try and show some of that data, is that with this technology, there's also the observation of a significantly reduced immune response to the antibodies, so we can generate very, very low levels of antibodies using this particular technology. Now, over the last several years, there are a large number of different so-called gene delivery vector systems that can be used to transfer these genes into the different organs in our bodies. But I'm just going to show you the data that we generated with one such gene delivery vector, and this is called the Eleanor-associated viral vector, or AAV for short. And you've probably heard about this in previous meetings, but the reason why we and many others are pursuing this particular gene delivery vector system is really born of the observation that it's not known to be pathogenic in humans, which is an important factor. But importantly, at least as compared to other viral-based vectors, it's significantly less toxic. And importantly, it's been shown to affect very long-term expression of our gene of interest, in this case is GAA, to treat Pompe disease. Now, this is an example of an experiment that we would generate typically if we built such a viral vector encoding for our enzyme GAA, and if we introduce this virus into the Pompe mice, we can show, as you can see here, very high levels of transduction, expression, and secretion of the enzyme into the blood, and that the secreted enzyme is taken up by all the Pompe-affected tissues, such as the diaphragm, the heart, as illustrated here, and that this expression is sustained for a very long period of time, in this study for 6 months, but we have others to suggest that these levels can go on undiminished for up to a year, at least in the mice. And that importantly, associated with these very high levels of enzyme, we can show very rapid and almost complete clearance of glycogen from all the tissues, essentially by 10 weeks post-treatment. And again, if you don't like the biochemical representation, I'm going to show you again the physiological sections of these animals. Again, from the heart and the quadriceps, showing very high levels of glycogen in the untreated animals, and following gene therapy showing almost complete clearance of glycogen from all these sections. As with the enzyme replacement therapy, we also asked the consequence of this clearance on motor function and coordination using the rotor-rod tests. And just concentrate on the rocking rotor-rod test. Again, yellow shows unaffected wild-type animals perform quite well. And again, the red shows the untreated Pompe mice showing a loss of function over time. And again, with gene therapy shown here in the orange and the brown, we can show maintenance, in fact, function that's very akin to what we see in otherwise healthy animals. So these animals were treated very early, and when they're around 2 months of age, we can show that we can completely correct the motor function and coordination in these mice. And similarly, if we look at the wire hang test, look for muscle strength, we can show again, in contrast to Pompe mice that show a loss in muscle strength, with the gene therapy treated animals we show performance. That's very similar to what is observed in otherwise healthy animals, at least those animals that are treated with a high dose of virus. Now this is obviously very encouraging, showing significant correction when we start treatment at the age of 2 months old, which is very, very young in these animals. So we ask next, well, what happens if we treat much, much older mice where there's significantly more pathology already and significantly higher levels of glycogen, as Nita pointed out? And this is, again, a repeat of the experiment I show you, but now the only difference is I'm putting it into very old mice, around 12 or 13 months old. And if you look at the enzymes that's made, we can do gene transfer in these old mice and see very high levels of production of enzyme and secretion into the blood. And we can also demonstrate, as we saw in the young mice, we can clear the glycogen from the heart and the diaphragm, although in the skeletal muscles, the extent of correction is less than what we saw in the young mice, and maybe in agreement with what Nina is suggesting, in that there are certain sections, at least in the older mice, that appear to be more refractory to treatment. However, despite this very encouraging clearance in glycogen, when we went ahead and measured motor function and strength, we were actually, well, I'm not sure to say disappointed, but we were actually surprised that the improvement that we saw were actually very modest compared to what we saw in the young mice. So clearly, although we're seeing some improvements, the extent of correction is much, much less than what we saw in the younger animals. And again, making the point that I think you've been hearing throughout this meeting, the importance and perhaps the important consideration of early intervention in the management of patients with Pompe disease. And to try and understand a little bit more about why these older animals were not responding, we had a look at the histologic sections, the muscles on these mice. So what I show you here is the section from a normal wild-type mouse. And I want you to notice that if you look at the nucleus, which are the dark spots in these muscle cells, most of them are located in the periphery, which is very typical of normal muscle cells. Occasionally you see a nucleus that's located what we refer to as the center of the cell, and that's usually indicative that there's some degeneration and regeneration has occurred in this muscle. Now if you compare this normal to the Pompe muscle this is from a 14 or 16-month-old Pompe mice, I hope you can appreciate that not only do we see the accumulation of glycogen, which you can see here as white punctate spots in this section, but also if you look at the location of the nuclei, most of them are centrally nucleated, suggesting that in this section of muscle, there has been extensive degeneration and regeneration that occurred. Now, if you look at this section, this is a mouse that we had treated by gene therapy starting at 2 months old. And not only had we removed the glycogen, but most of the nuclei, as you see here, is like the normal wild-type animal, that most of them are located in the periphery. And in contrast, and that may account for why we couldn't rescue function altogether, if you look at the older mice, if you treat them at 10 months of age, we can clear the glycogen to a very large extent, but a lot of the nuclei are centrally located, suggesting that perhaps one reason why we were not able to completely rescue function is because perhaps the regenerative potential of these muscles is no longer possible. So we run out of the cell-like cells that some of the speakers earlier mentioned that will allow for regrowth of these muscles. So again, making the point of the importance of early intervention as a consideration for management of Pompe. We also stain these sections for evidence of fibrosis. This is a permanent change in the architecture of the muscle. So again, this is a normal muscle. This is one from a 16-month-old Pompe mouse. And if we stain with trichome, that stains for presence of collagen, which is indicative of the onset of fibrotic lesions. We can show that there are sections in the muscles of these Pompe mice that show these indication. If we treat the animals very early and catch them early, we may never develop these fibrotic lesions. And again, if you treat the animals late, we can again rescue the glycogen accumulation, but there are already evidence of fibrosis in these muscles. Finally, I want just to finish by showing you yet another virtue of gene therapy, and that's what interests us in developing this technology further. And as I alluded earlier, is the possibility that this technology can confer what we refer to as immune tolerance. And what that means is that we can engender a much lower level of antibodies using this technology. So what I show you here is an example of an experiment where we compared the immune response of a mouse that had been either treated or not treated by gene therapy. So it's shown here in orange. If these animals had not been treated by gene therapy, and if you administered an enzyme, and we all know you would expect a generation of antibodies to the enzyme, as illustrated here, as indicated by the high titers of antibodies illustrated here. Now, if you treat the animals beforehand with gene therapy shown here in red, and do the same experiment, if you challenge them with enzyme, we can show that we do not generate significant amounts of antibodies. And these antibodies that we see actually return to basal levels very, very quickly. And you can re-challenge this and see no further induction, suggesting that these animals have acquired immune tolerance to the human enzyme. So in closing, I hope I've been able to illustrate to you that indeed with gene therapy, we can effect sustained and high levels of expression of this enzyme, and that this translates to near complete clearance of glycogen from all the affected tissues, and that efficacy was greater in younger than in older mice, as illustrated by the fact that we can see greater clearance, and that early intervention led to decreased muscle fiber degeneration and fibrosis, and significantly improved motor function and coordination. And that with gene therapy, yet another virtue is the ability to confer this so-called immune tolerance, leading us to again propose that gene therapy may indeed represent yet another way of providing essentially enzyme replacement therapy. And I'd like to end there, but I'd like to first acknowledge my co-workers, in particular Yongxiang Zhu and Robin Ziegler, who generated most of the data that I shared with you this morning, and also our collaborators as listed here. Thank you for your attention.